Yeah, let's get started. I got my little snack with my smoothie. Let's go. So I wasn't able to find a paper, and when in doubt, or when you just can't find something, phone a friend. So I called um, one of my friends and colleagues, which I pictured her here because she's actually on this paper. But I asked her, does she know any good papers? Typically, I try to pick a paper in advance because I always know we have journal club, but here we are. So I chose this paper, and it does match within the parameters of mitochondria, metabolism, and the cell biology stuff. So here we are. So I started this presentation sooner. I have like my title slide because HeLa cells are used in this paper, and I said that from now on, all paper presentations that I do that use HeLa cells or anything that was done by a Black scientist, I'm featuring, or just a Black person, period, I'm featuring that person. So, and this slide here is like a gearing slide. I always do a first slide where, I just put like images that relate to the paper so that it can kind of get people in the mindset of what we're going to be talking about in the paper. And so this is to let them know that there are HeLa cells in the paper, and these are the structures of NAD and NADH. And what I'll do is when I do the presentation, I'll ask, so hey, can anyone tell me what these structures are? Or, who do you think this person is? And then, that's how you get a dialogue in the beginning of the presentation. Then the next slide is the paper itself. And then also continuing the theme of highlighting Black people in science. I have a picture of Dr. Melanie McReynolds. She also graduated from my program, and she attended these journal clubs. So if not the newer students, a lot of the faculty will know who she is, and so I like her. I always like to tell why I chose the paper, and then I give a spoiler alert. So now we're going to actually get into the paper itself, where this is where I refer back to the paper so that I know that I'm saying exactly what the paper is trying to convey, and also so that I'm clear on what the paper is saying and if I actually feel like what the paper is saying is valid or well justified. That's a better way to say it. And so this slide here predominantly comes from the abstract. This is the abstract. Now, I may not include all of these methods at the end. It really depends on what specific paper figures I decide to emphasize. I might need to turn on the heat. It's getting a little chilly. So I have already started going in and copying and pasting some of the figures. And for now, I'm just gonna copy and paste all of the figures and then pick and choose specific ones out of it. So like right now, that is a lot of figures to talk about. I'm not gonna leave all figure A through J on one slide because that's too much to look at. You will see at the end how I break them up. But I think that those may be the figures that I choose to use because they make the most sense. Actually, I may go ahead and just take this one. So the way that I get my figures is I use the snipping tool. And I don't necessarily mind about there being that article review in the background. So then we're going to copy. Do a new slide. Click there, Control V, paste, and boom, there it is. So that is, the snipping tool is a great function. I'm not sure if like Macs have the same thing, but I definitely use the snipping tool in my PC. But if they better explain whatever the paper's trying to say, then I may use some, like one of the figures that I took came from the supplementary. This one right here, it came from the supplementary. That's because I just like metabolomics and I like looking at metabolic data. So I may also take this picture here. Not sure yet though. So this is enough to start with for now. Now I'm going to come back here and see if I can tell the story of these findings identify the SLC25A51. See if I can tell the story of these findings identify the SLC numbers as the first transporter capable of importing NAD into mammalian mitochondria. So that is the claim that they're making. And so this paper should tell the story through the figures, or I should be able to tell the story through the figures that support that claim. And so now I'm going to read through and figure out without, and I like doing it without the captions. I mean, there are some times where you really just need the captions to understand, but on the initial, I like to see what can I figure out about the figure without looking at the captions. And the figures where you can do that and like you understand it, those are the best figures. So note for when you're writing your own papers. So if I look at figure A, the figure legend, let me just make this bigger for now. So the figure legend says NAD plus, and it's looking at, and it's measured in nanomoles per mg in the mitochondria specifically. And then this is 293T mitochondrial NAD. So I don't know what 293T is. I'm assuming it's a type of cell line, and we're only looking at the NAD plus in the mitochondria. So I like to make notes of what I need to know, like what I didn't know going. So I don't know what 293T is. And it seems like they labeled CTRL is the control. And it seems like they labeled their shRNAs just by 1, 2, and 3, which doesn't tell me much. But I mean, in the grand scheme of things, you really don't need to know what they are, but I would like to know what they are in thinking about, the future of it. And so what I get from this panel A on figure 1 is that the control, there's about 3 nanomoles per mg of NAD in the mitochondria, whereas when you knock down any of whatever shRNA 1, 2, and 3 is, you have a significant, and that's what those 3 stars indicate, significance, which means that, this happens. And I think they have, what is 1, 2, 3, 4. So they have 4 replicates that they did, or 4 experiments that all support, that all went into determining the significance. And so significance means basically that this is very highly likely to occur under these conditions. And so it's not like, they just got this data as a fluke, or, it just kind of happened in a 1 in a million chance. No, when you knock down this shRNA1 or RNA1, you are going to get this response. And so in this case, you have a significantly less amount of NAD in the mitochondria knocking down all 3 of these. More predominantly, I see that in the shRNA2, you have the most knockdown, so whatever that one is. Looking at panel B, here they're looking at the NAD per mg of protein in the 293T whole cell NAD. Now, just off top, I'm asking myself why in panel A, they were looking at NAD measured. Oh, okay, now that makes sense. So they're looking at the per nanomoles per mg of the mitochondria versus here, they're looking at the whole cell. And so this is why they're looking at the protein. And so in the whole cell, you have more NAD, or at least from what these graphs are saying is you do have more NAD in these knockdowns, but it's not statistically significant. And so when something isn't statistically significant, you tend to think that it's just, it's comparable to control. Even though looking at this, I would say that it's a little, the shRNA1, I would say that's a little bit higher. But here, their statistics, it did not come out as statistically significant. And so I'm pretty sure that the claim that they're going to make from this, just from these 2 panels, is that when you knock down these shRNAs in the mitochondria versus the whole animal, in the mitochondria, you get a significantly decreased amount of NAD present, but there is no impact in the whole cell. So on a broader scale of things, I think that this would kind of indirectly tell people that just because, you may be measuring something within the cell does not mean that it is not impacted within a specific organelle within the cell. And so looking at panel C, we have relative fluorescence, how it's sensed. And here we're looking at the HeLa cells And we're looking at the free NAD within the mitochondria. And so here they only looked at now they're looking at the siRNA. And so yeah, we would need to know what and it may be possible just to know the difference between the siRNA and the shRNA, which short hair versus, I think short hair versus silencing RNA. I can't remember. I'm gonna have to refresh myself on what those stand for. But essentially this is saying that both of the SI1 and 2, both of them are statistically significantly decreased compared to control, but SI2 is severely affected, or SI2 severely affects the mitochondrial levels. Okay, so whatever these SIs are, and SIs and SHs are, they are impacting the levels of NAD. So we're seeing changes going on. Cool. And so now I wonder what made them, like, what was the jump between using these siRNAs and shRNAs to using the specific SLC 25A3233, 3651, and 52 genes. And so these are probably knockdowns, with the vector being the control. And I'm assuming that the NDT1 is a positive control, where we expect there to be greater than normal fluorescence, just so that you know your experiment is actually doing what it's supposed to be doing. So I feel like they probably got these from a screen of some sort. So when I read through the paper, we'll figure that out. But SLC 51 and SLC 52, they are comparable to the positive control, or they have statistical significance. So that's where, and if we go back to the title of that paper, it is SLC 25A51. So that's the bar in blue here. So I would think that they did some more tests and figured out something as to why they ruled out 52. We'll figure that out later. I'm gonna still put that question down here, but I'm pretty sure it's gonna be answered as we go through. We wanna know why they ruled it out, why and how, or how and why. Okay. Getting back to it. Okay, so that first panel of figures, that's definitely going to be in the paper. I mean, in my presentation, because I don't know, maybe not A, B, and C, but definitely D. We'll see. And so then they look at the emission ratio of NAD snippet. I hope that very well maybe pronounced snippet, but I have no idea what snippet or snippet is. I had already started putting notes there. Boom, back to this one though. So, and then they give you the wavelengths at which, so this here's the wavelength at which they were viewing them. And once again, they're looking at the mitochondria, the free NAD in the mitochondria. The control is here, and the knockdown I don't know what they've knocked down, so I need to figure that out. But whatever that is, it is significantly decreased. Okay, and then they do the same thing again here, and they look at the U2. Oh, okay, so here they're looking at two different cell types. So that's the HEK, that's the cell type, and the U2OS. So I've heard of HEK, which actually, so that may be what the 293 is, because that says 293 here. I don't know, but I have to check that out. And then U2OS, I have not heard of U2OS, so I'm gonna have to figure out what that is as well. And then you continue on here, and when you look at the free mitochondrial levels, so I'm wondering the relative fluorescence of free mitochondrial levels. So I'm guessing, actually, I might have explained this figure wrong, because here there is a increase in fluorescence in these animals. So I'm thinking they're measuring the free, they're measuring the mitochondria. Yeah, see, this is why it's important to know what methods they use and what they were exactly measuring, so that you can know what the figure's showing. But this is just my go through to see what I can see. I'm guessing this is measuring the mitochondria, and in the knockout, the NAD is significantly decreased. I wonder if they like overexpress. This might be some overexpression experiments where they overexpress. I don't know, we'll see. Wholesale, so this matches up with this. So the HAP1 cell, you look at NAD levels in the knockout, really no changes. You look at the NAD levels in the knockdown, because shRNA has a knockdown, and you see no real changes as well. So those two match up. This whole graph, or this whole figure, is just to indicate that there are differences in the mitochondrial levels and differences of NAD levels within the mitochondria versus the whole cell. And they use a number of different cell types to show us that. And I think that this is convincing. So step one, and then like I'll come and I'll come and actually title this as, and so this is a temporary title, but what I would title this as is NAD levels are impacted, NAD levels, something to say that like NAD levels are different. This is a very temporary, different in mitochondria compared to whole cell under specific conditions. Something like that, just so that I know what that figure is about. And then I will go and do this for all of my other slides. So I will check back in once I finish doing that. Oh, y'all, I am tired. I'm not tired, but I just need a break. I've been working on this for about 2 2 hours, an hour and a half-ish at this point. So I just want to show y'all like some of the decisions that I made and what I did. So this is where we left off. And I actually decided, and I didn't do it here, so I'm going to do it really quick, but I decided that I'm going to cut off these last two, those last two plots there. And I need to read a little bit more to figure out which of these figures I want to keep. So we're just going to keep that as is. But then I added a additional slide where we're only looking at the metabolic changes in the whole cell versus the mitochondria. And so I have two representations of what that looks like here. One of them is a heat map and the other is a volcano plot. I'm still trying to figure out how I'm going to show these because I thought that I was going to like it the way it looks here, but I don't. So I'm probably going to do a bit of rearranging of some sort so that like these two figures are large and clear. Yeah, I'm not too sure yet, but we'll figure that out. But I do want to have a slide in itself where I look at the mitochondrial changes. I'm sorry, when I look at the metabolic changes, which is looking at the mitochondrial changes. And then I really picked apart this figure. It's kind of easy because the point of the figure was obvious in that they were saying that mitochondrial function is decreased when whatever gene they're knocking down and knocking out is decreased. And so I want to emphasize here that the overall point and what, like the title is not saying exactly what the figure is saying. It's me taking that extra step forward and then interpreting what the data is saying and putting it in my own words. Now, of course, I still have not fully read through the paper. So we're going to see how well my conclusions and interpretations of the data match up with what the paper's conclusions and interpretations are. But what I got from this, and I'm going to make it big screen, what I got from this is that every figure had these two, every figure had these two figures. So the figure on the left, this one here, this is looking at the oxygen consumption rate, and it's showing you the actual graph, the respiration rate as a function of time, right? So this is the basal, this is from time 0 to 20 minutes. And as you can see, automatically off top, the knockdown, in which over here is the figure legend for this and this figure, but the knockdown is significantly lower. And I can say significantly because here you see those two stars. And then you come and you look at the maximum respiration and you see once again still it's lower. And so these two figures are telling you the exact same thing. It's just showing you the visual. The bar graph is an easier thing to visualize, and it's an easier thing to interpret. And so, if you wanted to view it this way to see the function of time, sure, you can do it that way. But for quick and ease of interpretation, I chose to, because they did this same experiment 3 or 4 different times. And so for the rest of these, I removed this oxygen consumption rate line graph, and I just included the bar graph that's really gave you the concise summation of the information that you really need to know. In terms of this one here, I chose to show both graphs because looking at this bar graph, it gets a little bit confusing in that you tend to want to compare everything to control. And when you're going 3-4 bars out, it's, you can still compare to control, but I really like this visual And once again, when you're doing these journal clubs, you get to decide what visual you think looks best and explain why. That is an opinion, right? So someone may feel like, oh, this is still the best visualization. Whereas for me, I think in this case, when you have multiple bars, the going back to the line graph is a better visualization, because you can very quickly and easily see that, oh, there's one that's right at control, like it follows control. There's one that is below control when it comes to the max, but then at the basal, it's right at, the same level as control. But then you can see that there's also one here that's always higher than control. So I appreciate this graph over this one, but I still want to show both of them, especially considering, the colors, and for those who may like that one better. And I need to read a little bit more to figure out if I really want to include this portion of the figure, because I'm not completely sure of what I'm looking at here, but I have to read a little bit more. I did not get to this figure, and yeah, so I'm going to take a break and come back to this, and I will give y'all another update. An update on the presentation. It ended up being a total of 13 slides, as you can see here. And I'm just gonna quickly go through and tell you what I did and briefly why. So I decided against my traditional gauging slide that I had talked about earlier, and I just went with a fall themed slide. This here is my introduction to the paper, and I have highlighted down here the key takeaway from the paper. And I also made a comment in noting that I really appreciate how clear and specific the authors decided to present their findings. And then, yes, I did still highlight Dr. Melanie McReynolds, which I will link the conversation that I had with her in the top corner here. And so I completed my spoiler alert slide, and this is just basically to orient everyone before we get into the paper so that they know what exactly the paper is about, what they should expect, and so that they're thinking about the experiments in the same way that I'm thinking about them as we're going through the paper. And it really does help to create a more engaging conversation because everyone is on the same page and they are all looking towards the same end goal. And I didn't say this earlier, but I would also like to note that in the known space where I talk about some of the known information, I did have to look at how they know, quote unquote, know this information. And that typically requires looking at the papers, the groundwork papers that are used in this paper. So whenever you're doing a journal club or presenting a paper, you are going to need to talk about review not only the paper that you're doing, but then also what paper led up to this paper. I decided to talk about all of the different cell lines and their origins. I found it quite interesting in seeing where the different cell lines that were used came from. I actually got into a conversation about the vagueness of knowing who these cells came from. And it's actually a protection for the family because these cells came from people who did exist within families. And so let's say that we go and sequence these cells to sequence the DNA from these cells. We are now be able to understand someone else's genome, and then that becomes public knowledge. And so that's the ethical issues and things that I had a conversation with about identifying where these cells came from, like the actual person versus leaving them anonymous. And I found that interesting. So this was my introduction to the paper, the figure slide. And as you can see, I just have a panel of 4 images or 4 graphs. And my title here is very clear in saying that NAD levels are different in mitochondria compared to whole cell. I did end up just giving the 2 different metabolomics or metabolic response figures their own slide. And I talked about them individually. I also broke up the figure that was looking at oxygen consumption rate a little bit more because there was a lot going on in that figure as well. And I added in a couple of other figures, and I included this last figure here because this was the figure that the authors felt like really drove home the message, gave the conclusion that they were looking to give. And so we spent some time as to whether or not this was the bread and butter figure of the paper. And then I have a conclusion slide, and that was it.